2018 past paper, starting the multiple choice. Okay, which row in the table describes properties of proteins that allow them to be separated using the 10x shown? Pretty much need to just know these ones. So centrifugation is when you're going to put them into a tube and then you're going to spin it around really fast, and the heaviest stuff gets pushed to the bottom and the lighter stuff stays at the top. This is to do with density. Okay, gel electrophoresis is where you put it into a agar gel with wells at the top, and then we basically give it a shove with a negative charge at the top, so we've got an electrical current moving through like this and it will move down. So this is to do with charge, and that's why that's E. The three-dimensional structure of a protein is shown. Which row in the table best describes region X? So we have region X here. Well, there are two forms of secondary structure. You have an alpha helix, which is a twirly thing, and you have beta pleated sheet, which is an uppy downy thing, which we show as a kind of flat head arrow. So what are we seeing in region X is twirly, so we are alpha helix, and then we need to know what stabilizes secondary structure is hydrogen bonds. Peptide are the primary structure. So the diagram below shows some interactions between amino acid R groups in a polypeptide chain. Which letter shows hydrophobic interactions? Okay, so I need something which is basically things that don't like being near water. I am looking for non-polar to non-polar. Okay, that's really, let's find something like that. Okay, let's get rid of this one because this is ionic. Okay, we have charges and they like being near water, they're okay. Let's get rid of this one because this is covalent. This is a disulfide bridge between two cysteines. And this one here is again charged, associated bit with the hydrogen coming off here, so that's a hydrogen bond. Also super happy to be near water. This, however, these are both non-polar, so the correct answer is B. Okay, a little bit sums on this one. Okay, so you could do this lots of different ways. We've got a student preparing media for an experiment to investigate the effect of an inhibitor on cell growth. Flask 1 contains a control medium with no inhibitor. The concentration of the stock inhibitor solution used to prepare the final solution is 80%. You are asked to work out what inhibitor volume and what buffer volume are required to make up this total thing. Okay. What volumes would be added to Flask 2 to give 25 centimetres cubed of medium with an inhibitor concentration of 20%? Right, I did this in two stages. So I looked at this and I said, right, okay, my medium in total that I already have, I've already got a 1 and a 5. So I already have 6 centimetres cubed, and I've got to make it up to 25. So I know my total that I can add is 19 centimetres cubed. Now this helped me because straight off, here's over 19, and this added to this is well over 19. So that got me rid of these, and I just stopped kind of panicking about what I was looking at. Okay, so now I needed to make a decision between these ones. And so what I now looked at was said, if it is originally 80% and I want to get to 20%, that's a 1 in 4. Okay, because I need to dilute it by 4. So I am looking here at a quarter of 25. So a quarter of 25 from a total is 6.25, and that's how I got my answer. Okay, question 6. Transcription of gene Z only occurs when its transcription factor is dephosphorylated. D. That's probably helpful. The distribution of the transcription factor together with the activities of a protein kinase and a protein phosphatase specific to this transcription factor are shown in the table. Okay, so being very careful about what they're doing. Which one? So it's saying gene Z is transcribed in, and we've got muscle, heart and brain. Well, for a start, if there is no transcription factor, then the muscle's just not going to get any. So we'll take that one out straight away. Okay. And so therefore, these guys are in with running, and we just have to decide between the two. What do I need to have? Kinase or phosphatase? Okay, so kinase transfers phosphate from one place to another. Phosphatase removes phosphate from something. And this says only occurs when its transcription factor is dephosphorylated. So basically, I need this. Hence, it is the brain, and so E. Okay, another sodium-potassium pump question. So again, I've gone and got the picture just to help. Okay, so this is our internal, and here is our external. It's not the nicest question. They're asking you to really know what you're doing. I can't spell external, excuse me. That's better. Okay, the effect of changing the concentration of extracellular potassium ions and the function of the sodium-potassium pumps was investigated. Starting with an extracellular solution containing no potassium ions, so I've got nothing, I have no K up here, okay? And then I'm going to increase K, so I'm going to start adding in some more. As the concentration is increased, what would happen to the pump? Okay, right, so for a start, can we get rid of A and C, because these are both incorrect, because what you're doing is pumping out sodium and in potassium, so A and C are not in it. And your options are either stay in their phosphorylated conformation for longer or hydrolyze ATP at a faster rate. So in its phosphorylated conformation, okay, that's when we have in this section here, so our sodiums have attached, we've then phosphorylated, which has changed the shape, and the sodiums leave. Okay, it's only at that point that the potassiums combined, but once the potassiums have bound, it then undergoes dephosphorylation. So the more potassium I have available to do this binding, the quicker it's going to do that. So what we're going to get is hydrolyzing ATP faster. Otherwise it would just be stuck waiting for potassiums to click in, which is what we have for B. Okay. Right, a sample of 10 to the 6 cells was found to contain 0.4 mg of protein. Actin comprises 4.5% of the total protein. 42 grams of actin contains 6.02 times 10 to the 20 molecules. 1 gram equals 1000 mg. The number of actin molecules per cell is, it's just number crunching. Okay, so just don't panic. So what we have is 0.4 milligrams. Okay, so what that really is, if we take 0.045, because it's 4.5% of that, that would tell me that in reality I have 0.018 milligrams of actin. Okay, what I then did is just a straight proportion. So it tells you 42 grams, so that's 42, use the conversion, 1000 megs, okay, gives us 6.02 times 10 to the 20 molecules of actin. So now I plug in my 0.018. Okay, straight proportion, so this one divided by this one times by that one, okay, gave me 2.58 times 10 to the 14. Okay, now unfortunately this is an answer, they have been horrible about this. So this is not though per cell, notice this is in bold. This was a sample of 6 times 10, sorry, 10 to the 6, not 6 times 10. Okay, so what I need to do now is divide this by my 10 to the 6, so I can get back down to just being 1. So what I have is 2.58 times 10 to the 8. Okay, which row in the table describes the states of the proteins P53 and RB that would increase the rate of cell division? Okay, so P53 is a tumour suppressor. So I'm looking for something that is increasing the rate of cell division. So I need this to not be doing what it does best, which is to stop cells dividing. So I need this to be inhibited. Okay, and then I've got my retinoblastoma. Okay, this one is what you're looking at your G1 checkpoint. So what I need here is I need this to be phosphorylated. So that is two very specific points of data that you need to know for you to be able to get the answer, which is B. Which diagram shows the correct positions of the cell cycle checkpoints, a checkpoint represented by this line, thick line? Again, it's just something that you need to know. There are 3 checkpoints. Okay, there's a checkpoint for going into S phase, which is at your G1. You then have a checkpoint once you've come out of it to make sure that all your synthesis has happened at G2, and then you have a final kind of commitment issue. You got to check that everything's okay at mitosis. That's the final one. Once you get past that, you're committed to division. Okay, so I'm looking for a G1, a G2, and an M, and so look at what you've all got here, and it's just here that you have that. Okay, question 13. In a study of transmission of the rabies virus by vampire bats, charming, the density of bat colonies was estimated using mark and recapture. Total population estimate is given by MC over R. So this is actually a formula that you're expected to know, but sometimes they give you. Okay, so what we have here, this is our first sample. Okay, this is our second sample, but just literally how many we actually caught. And this is the second sample that was caught and was already marked. So this is your recaptured section. Okay, so this one, we're seeing that they've already worked out the population at 440. And following the capture of a second sample of 64. And so what we're trying to work out is M. So M times 64 divided by 8, because this is how many were marked. So if I rearrange this just for ease of things being in the right place, okay, what we're looking at is M is 440 times by 8 divided by 64. Plug that into your calculator. There is your answer. Which of the following describes the purpose of a randomized block design? Okay, so a randomized block design is not about ranking. It's not about representative sampling, because that is to do with randomized sampling, I suppose we're looking for here. It is not about monitoring the dependent variable, because that is to do with your design in terms of your sampling at the end of it. Okay. However, it is very good for controlling for confounding variables, other things that might be causing an issue that you're not ready to, like if it's like a field trial one, you're looking at it being on a slope, so you would randomize for that. Okay, question 15. Merlins are small birds of prey which chase and capture skylarks. The effect of skylarks' song on hunting by merlins was studied. Graph 1 shows the number of successful and unsuccessful attacks, showing different singing behaviours, and graph 2 shows how the different singing behaviours affected the mean duration of chases by merlins attacking skylarks. I'm just going to move this down so we can actually see the question set here. Okay, which of the following generalisations about skylark singing behaviour is valid? Okay, no song decreases the number of attacks by merlin and the time they will chase. So here is my no song. Okay, decreases the number of attacks. Well, here's our full song, there's our sub song, there's our no song. In total, I would say that we have a decreased attack. Yes, decreased number of attacks. And the time they will chase. Here is the chase time for no song. Well, that's not true. They're chasing them for quite some time, so it's not A. Okay, no song increases the success of attacks. So, and the time they will chase. So here we go. Here's our successful attacks. Full song, sub song, no song. Yep, I agree with that one. And the time they will chase. Yes, we agree, because that one is big. Just to check the other two. No song increases the number of attacks by merlins and the time they will chase. We know the time they will chase is correct. The number of attacks, though, no. I mean, like this is much bigger if I add these two together in the sub song, and probably the full song as well. The no song, even adding these two together, that doesn't stack up. No song decreases the success of attacks. Well, we've got that as here's our successful attacks getting bigger, so it's not that one. And it is the time they chase. Okay, so it's definitely B.